And then about 5 or 6 days after fertilization, the process of hatching takes place. I imagine you probably didn't realize that humans hatched, but it's in a very different sense than a hen might hatch from an egg. Nonetheless, what has to happen is that this trophoblast surrounded embryo has to emerge from the zona pellucida. What happens is it actually pumps some fluid into its inside, and as it expands, eventually the zona pellucida will rupture. And in the picture in the extreme right, you can see that the developing structure, the embryo itself, is beginning to hatch out to emerge through the zona pellucida. Now, this is an important stage, and if this was to happen prematurely, as we'll see, then there can be difficult consequences as a result. And these pictures, incidentally, are from an in vitro fertilization laboratory, and this is an individual who may well have developed into an adult at some point. And now we're moving to a diagram. It's often convenient to use diagrams rather than photographs, because in the diagram you can colour things and make it easier to follow which cells give rise to which. So here in this diagram, I've coloured the original inner cell mass is red and yellow. And that's to indicate that it's divided again into another 2 different sorts of cells. So the cells shown in red are primitive ectoderm cells. The cells shown in yellow are the primitive endoderm cells. But you can probably also see that the cells of the trophoblast have distinguished themselves into 2 different sorts as well. The cells from the original trophoblast round the outside are now known as cytotrophoblast cells. But you can see that up at the top, where it's beginning to implant into the uterine wall, then the cells have lost their cell membranes. So you have nuclei, which are effectively freely distributed within one mass of cytoplasm. And this is called syncytiotrophoblast. Now, it sounds a long and complicated name, but it's doing a very important purpose, because it's a syncytiotrophoblast that will help the developing embryo implant into the uterus, into the wall of the uterus. And as you can see in the picture, it's beginning to kind of invade its way in, to eat its way in to the underlying tissue. You'll note in the uterine tissue, I've also sketched in some blood vessels surrounded by endothelial cells and with red blood cells within them. And of course, the uterus is also rich in uterine glands. So at this stage, the total overall size is still very much like the original fertilized egg. Growth will only start to take place as it begins to receive nutrients from the mother, from the uterus. And we're about 6 days after fertilization at this point. The inner cell mass, as I say, has given rise to ectoderm and endoderm, and it's only a few of the primitive ectoderm cells that are going to give rise to the baby that will eventually be born. So you can see that at these early stages, most of the cells will contribute to the support network, to the placenta and support structures that will help the embryo and then the fetus develop along the way that it ought to do subsequently. So these can be seen as the support cells for the developmental process. Here we have 2 days later, about 8 days after fertilization. And now the whole pre-embryo is beginning to disappear under the surface of the uterine wall. So it's almost invisible from the outside. There's perhaps just a little scar, just a little plug of tissue, if you were to look inside the uterus where the implantation process has taken place. But otherwise, it's burrowed its way under the surface of the uterine wall. And in doing so, you can also see that it's engulfed some of the blood vessels that were present in the wall of the uterus. In fact, it's actually effectively dissolved the walls of these blood vessels, so that the maternal blood, the red cells shown as red dots in the diagram, are now directly in contact with that tissue that we call the syncytiotrophoblast. Our red cells, the primitive ectoderm, they've also begun to hollow out. They've begun to form another hollow cavity. And that cavity is called the amniotic cavity. And that will, in fact, be the amniotic cavity of the later developing baby. So it will become very large and significant subsequently. Equally, if we're to look at the yellow primitive endoderm cells, they're beginning to move round the inside of the trophoblast shell, and eventually they'll meet and they'll form another structure. So we can look at that in another diagram, and here you can see the primitive ectoderm is forming the amniotic cavity, and the primitive endoderm is formed this yellow, marked in yellow, endoderm cavity closed over, and that will be the yolk sac. It's called the yolk sac as an evolutionary marker. Long ago in our evolutionary history, when we were still developing inside eggs, then we had yolk. And it's the process of evolution that moved us away from being inside eggs to being inside a mother. But we still have a yolk sac. So it's still a marker of our long evolutionary history. If we look at the blood cells of the uterus, we can see now that they have been entirely engulfed by this syncytial trophoblast. And in fact, some of the trophoblast cells shown in the diagram in dark brown are beginning to develop finger-like projections, or villi. And this is an early marker for what will be the placenta. But what they're doing at this stage is to increase the surface area across which nutrients can flow and wastes can be exchanged. So it's forming some of the functions of the placenta in this very early and primitive form. And of course, now that it's connected to the mother, then growth can begin to take place. It's taking nutrients from the mother so the whole system can begin to expand. And there'll be a large number of changes of arrangements and morphology before it will get to anything we can even begin to recognize as looking at like a human embryo. So at the moment, it would be almost impossible to spot where development is going to take place. This does not look like a human in any sense whatsoever at these stages.